There are two paths in the world. One is the sword and the other one is pen. But there is the third path stronger than both. That is a woman. Good morning, everyone. I, Dr. Sakshi Bhardwaj, welcome you all and much obliged to have you all here in the Women's Day special webinar series. That is, Women Researchers in Computational and Biology. It is just an initiative to highlight the contribution of women researchers in diverse fields to construct an innovative scientific world. We are fortunate to have such a special guest of speaker with us today who has her expertise in both biological research and photodynamic cancer treatment. She also gained substantial knowledge in several other related subjects, including optical imaging, diagnostic, and low level light therapies. It gives me immense pleasure to introduce our guest of speaker, Dr. Arpana Parihar. She is currently working as a woman scientist B at CSIR, Advanced Material and Processes Research Institute, Bhopal, Madhya Pradesh. She did her PhD from Raja Ramaiah Center of Advanced Technology, Indore, and postdoctoral from the Center of Biomedical Engineering, IIT Delhi. Dr. Arpana is awarded with prestigious fellowship like GATE, CSIR Net, DST, Women Scientific WOSA and WOSB. She has more than 8 years of research and teaching experience in the field of disease therapy and diagnostic. Her current research activity includes the fabrication of biosensors for the early diagnostic of cancer, molecular docking, and simulation for the drug designing, tissue engineering, targeting cancer therapies, and 3D cell culture. She has published 45 articles and edited 7 books with the renowned publishers such as Elsewhere's, Wiley, Springer's, etc. Today, Dr. Arpana will be talking about the advanced graphene-based nanomaterial for fabrication of electrochemical point-of-care devices for the cancer diagnostics, along with the fundamental of traditional detection methodologies that would pave the way for the next generation sensing methodology. So now, not taking much time, I would like to hand over to our expert of this masterclass. Over to you, ma'am.
Thank you so much, Dr. Sakshi, for such a nice introduction. And thank you so much for, I would like to thank Ultim Technologies for giving me this platform to present my work. And the today topic of my talk is advanced diagnostics for healthcare management. So, let's start. I will brief my topic under following heads, or I will give first an overview of detection of analytes. What are analytes? Why we want to detect them, and the conventional techniques which are available for the detection of these analytes, and why we need advanced technique, and what are the advantages of advanced techniques over conventional techniques. I will give a brief overview. Then I will talk about the components of biosensor, and specifically, since my group is working in electrochemical biosensors, in the field of electrochemical biosensors, I will talk about the electrochemical biosensors, their working principle, features of an ideal biosensors, some a brief historical overview from where these biosensors come from, and simultaneously I will also talk about some nanomaterials, and which is the most feasible strategy to enhance the biosensing properties. And along with this, I will shed light on synthesis and advantages of nanomaterials, and will give some examples of commercially available POCT biosensors, and then will brief about my ongoing research work over diagnosis of cancer biomarkers, and what are the biorecognition elements, how my research is going to advance the field, and finally I will conclude the topic. So I hope you all will enjoy my talk. Let's start with some basic. What are analytes and why the people wanted to detect them? Analytes are any molecules, they are any molecules or compounds which are present in environment, and at a certain level they are harmless, but after certain level they become harmful. The example of these analytes could be like, they can be pesticides, heavy metals, antibiotics, which may pollute our water, soil, air, and thereby causes environmental pollution, which affects the human health drastically. There are several types of screening and sensing technologies, or I can say the diagnostic technologies, which are available for the detection of these hazardous chemical or small molecules. And their detection is very much crucial in order to prevent the environmental pollution and thereby several diseases. Conventionally, there are several technologies. I put some monograph at the right hand side image, like HPLC, GCMS, LCMS. They actually they provide highly accurate results and quantitative results about the particular analyte. Um, Further molecular techniques are also available for detection of certain other kind of molecules or analytes. We can say them like microbial infection, infectious agents, bacteria, viruses, etc. And for detecting these type of bacteria, viruses, PCR, RT-PCR, and hematological tests generally have been employed. Some advanced techniques which have now been introduced nowadays, like LIFA, lateral immunofluorescence, CRISPR, CRISPR-based techniques, and biosensors. They actually, I will talk about their advantages over conventional technologies in next slides, where first will I will brief about the conventional techniques for detection of analytes or detection of biomarkers associated with a particular type of disease. Conventionally, as I told you that HPLC and GCMS and LCMS, they are generally useful for detection of analytes like pesticides, heavy metals, antibiotics, etc. But certain techniques such as CT scan, MRI, PCR, RT-PCR, hematological tests, they are usually useful for the detection of microbial infections or any kind of disease progression, such as like oncological disorders or several non-oncological disorders, arthritis, etc. These conventional techniques, they provide accurate results and generally useful in many cases. However, these techniques needs they have certain drawbacks, setbacks, such as they need centralized facilities, expensive bulky instruments, skilled personnel, and tedious sample processing uh, methods, which restricts their usage in remote low resource areas. There are certain advanced techniques which are handy, they can be miniaturized, cost effective, and can be integrated with several other platforms. Examples of such techniques are electrochemical biosensors, LIFA, CRISPR, Cas9 system, surface plasmon resonance, LSPR, and biosensors. These techniques actually, we will talk much more deeply about these techniques in today's talk. The biosensor based approaches are cost effective, easy to use, portable, and generally they offer a quick response time to us. They are highly sensitive and selective too. So, what are the biosensors? Uh, a biosensor is an analytical device for the detection of an analyte that combines a biological component with a physicochemical detector. You can see in this diagram that biosensor is just based on a principle where analyte interacts with its biological receptor, or we can say it biorecognition element also. They can be anything like enzyme, antibodies, nucleic acid, whole cells, or aptamers, um, where the where the analyte get bind, because they possess high specificity towards a particular specific analyte binds with these biocomponents or bioreceptors or biorecognition elements and produces a signal. Can be in any form, like it can be a optical signal, like we can see the color change or fluorescence, or it can the binding of analyte with a bioreceptor may cause a change in mass, which we can detect using highly sensitive method, piezoelectric based or acoustic based method. Or they can produce a magnetic response, or they can produce a current, which we can measure via amperometric or potentiometric electrochemical biosensor. Or they can change the temperature. The binding of analyte with a particular bioreceptor can change the temperature, so we can measure it via thermometric mode of measurement, where these biosensors can be utilized. They have wide application. They can be employed in clinical diagnostic procedures, environmental screening methods, and to assess the food quality. So overall, to sum up this slide, biosensor comprises mainly the two elements: a biocomponent or biorecognition element, or a signal transducer, which actually process the signal, amplify the signal, and give us the output. So this is again showing a simplified way what kind of like analyte can bind what kind different types of biorecognition element, which is very specific for a particular type of analyte. And so then biorecognition element can be MIP, that is molecularly imprinted polymer, antibodies, enzymes, whole cell can also be act as a bioreceptor, nucleic acid. They can be aptamers. Aptamers are nothing but they are single stranded or double stranded DNA or RNA molecule which have high specificity towards their analyte, and they can be phages. So based on the transducer part, the biosensor can be further subdivided into electrochemical, optical, and others. And electrochemical biosensing method can include amperometric, conductiometric, impediometric, potentiometric mode of analysis based on the measurement of like where we are interested, like we are in if we are interested in measuring current or in voltage or in potential difference, so we can subdivide these method. And biosensor can be of optical type, where we are interested in like measuring color, like colorimetric or fluorescence or plasma resonance or photospectrometric mode of method. They all comes under the class of optical. And others like thermometric, piezoelectric, FET, and microcantilever based biosensors. Biosensors are cost effective, they are highly selective, they provide rapid and early results, and so they are feasible for as a diagnostic technology for monitoring biological diseases. They can be disposable, like nowadays LIFA based like colorimetric sensors, they are available in the market. They are disposable, and they can be used as a in point of care settings near the patient bedside. So this is the overall how electrochemical biosensor, since my group is basically working in the field of electrochemical biosensor. So this is a layout how electrochemical biosensor looks like, or how it works. So we there is actually 3 electrode system. One is working electrode, another is counter, and the third one is reference electrode. The working electrode is one. It can be silver, gold, platinum glassy carbon electrode, or it can be a screen printed electrode, or ITO, indium tin oxide sheets. We can all these type of working electrode can be used depending on what material we are using for fabrication of our electrochemical biosensor. Counter electrode is generally made up of platinum, or it can be of any other metal, and reference electrode is AgAgCl. These three electrode system is connected with a potential state, and which is connected with a laptop, or we can nowadays it is available, we can connect it with a smartphone also, and it will give us this type of response like this curve is the upper hand curve is known as cyclovoltammetric based detection, and the lower one is Nyquist plot, which is obtained from uh, EIS, electrochemical impedance spectroscopic method. So this is overall the setup, and what is the working principle? As I already briefed you about the working principle, there is a transducer which can be electrical, optical, or physical. Over this transducer, our like this transducer basically transform the signal. What kind of signal? Signal generated by the process of binding of analyte over the biorecognition element, and this signal actually this input signal is amplified by amplifier, and we are able to detect it over the monitor. So there is a term biorecognition element. I hope you all are aware about this biorecognition element. As I earlier explained you that these are generally the enzymes and aptamers, MIP, or antibodies. They can be anything based on our experimental setup. We can choose BRE of a specific type. So ideal feature, what are the ideal feature of biosensors? Biosensors should be, it should be highly sensitive so that it can actually detect the analyte in a much more sensitive manner. Sensitivity actually is the value of electrode response per substrate concentration. It should be, biosensor should be cost effective. It should be highly selective. Selective means it can be able to detect our analyte in presence of different interference chemical. Okay, like if we wanted to detect the glucose in the blood, there are so many other analytes in the blood. Analytes, we can say protein, carbohydrate, fats, but specifically our glucometer is able to detect the presence of glucose. So this should be selective. The biosensor should be selective. The response time should be quick, and the shelf life of biosensor should be like long, so that it should not be like falling, or it should not be like poisonous or anything like that. And nowadays more advanced biosensors are available. They are multiplexed means one can able to detect more than one type of analyte simultaneously. There are several disorders such as cancers, arthritis, etc., which are associated, they're causative actually associated with a multiple types of factors. So if one can monitor more than one type of analyte in that diseases, in the case of that diseases, one can more confidently detect or we can say diagnose the disease progression, the way disease is progressing, and we can target, we can design the therapeutics according to the level of disease progression. So these are the some features for ideal biosensor. So let us talk about some brief history of about from where biosensor actually from their invention to the present. First time in 1954, Clark has invented oxygen electrode, and this electrode was first time able to detect the oxygen in its surrounding. Then later on in 1956 or 1975, first commercial in 1975 2025, we can say, first commercial biosensor is available, but it's a very, you can see in the picture, it's a huge size, and not at all portable. And nowadays we have much more portable biosensor, which are like integrated with the artificial intelligence, and they have revolutionized the field of disease diagnostics and therapeutics. With the advent of these artificial intelligence or smartphone integrated biosensors, the nowadays wearable sensors are also available in the market, so one can monitor the various parameters of body, such as glucose, lipid profile, etc., in a real time manner, so that one can check the health of a person in a more personalized way, rather than earlier it was like conventionally we have to go to the lab, and the lab were giving us the results into after 2 to 3 days. Until that, the patient may be like the disease get worst. So how from primitive biosensors, the advancement happens just due to the advent of these nanotechnological field, where the nanomaterials, they actually use to enhance the biosensing properties of the biosensors. Nanomaterials can be of various types, like graphene, nanowires, dendrimers, carbon nanotubes, quantum dots. What they do actually, nanomaterials basically, they enhance the surface area. They basically increases the surface functionalized moieties over which our biorecognition elements binds, and produces a signal when it reacts with the analytes. So in case of electrochemical biosensors, by employing carbon-based nanomaterials or any other sort of nanomaterials, one can enhance their sensitivity up to the femtomolar level, and the limit of detection can be enhanced, and one can exploit these as a promising tool for the various kind of disease diagnostic and prognostics. This is just a brief overview how we can synthesize various types of material, as it is in itself a vast field that there are various synthetic approaches to synthesize various types of biomaterials, and one can categorize them into 0D, 1D, 2D, 3D types of materials. Like in 0D, most of the quantum dots, fullerenes, they come, and CNT, carbon nanotubes, they generally comes under the class of 1D. Graphene, maxenes, and MOS2, they comes under the category of 2D. And there are 3D sheets like graphene, which can also be employed in at certain places for certain other application, energy or solar cell applications. There are mainly two ways: top down approaches and bottom up approaches for the synthesis of these nanomaterials. Generally, arc discharge, laser ablation, and implantation, and molecular surgery sort of things, people have used to synthesize these molecules in a specific manner, so that they can enhance a particular type of properties for which they are meant to synthesize. Generally, these nanomaterials, they enhance, they actually, in the field of biosensors or biosensing for biosensing application, they generally enhance the specific surface area of binding. These nanomaterials, they should be biocompatible, so that they can easily interact with the biological molecules. Their conductivity, like their properties, electrical, magnetic or optical properties, should be optimum, or one can say they should be enhanced, so that the signal output get increased, and we can detect much, the limit of detection should be like in enhanced. So and as I already said in my previous slide, that these nanomaterial provides various functional groups such as NH2, COOH, sulfhydryl group, hydroxyl group for the immobilization of biorecognition element. These nanomaterial, they generally they transfer the charge from conjugated backbone to the our transducer, and they should be, like in case of electrochemical biosensor, they should be electrochemically active. They possess high adsorption capability. Their electron communication property should be like electronic band gap should be optimum, and they can be like dispersed in both aqueous and non-aqueous solution. And if these all properties are feasible, we can exploit these nanomaterial in the field of environmental screening, biomedical, disease diagnostics, food quality analysis, electronic item synthesis, in the field of healthcare management, and in the field of industrial applications. So these are the some very well known and famous examples of commercially available point of care devices. There is first one is the most famous and most successful biosensor is the glucose biosensor, which detect the glucose in the blood sample. Here the reaction is very simple. Actually, it is the enzyme glucose oxidase is immobilized over the screen printed electrode, which upon reaction with the glucose oxidize into the gluconic acid and release H2O2. This H2O2, when interacts with the platinum AgHCl electrode, it produces oxygen, hydrogen, and electron. These electron amplified by the amplifier, and we are able to detect the signal. Similarly, the another famous is the P test, pregnancy test, where the human gonadotropin hormone is immobilized over the strip, and it can gives us a detection signal in the form of color change. And most recently, there are the COVID-19 detection LIFA based biosensors are available in the market, which are able to detect the COVID-19 antigen, or there are the biosensors which can detect the antibodies present in patient serum. So let us talk about the disease biomarkers. Actually, there are 4 to 5 types of biomarkers. Biomarkers are nothing but they are the measurable indicator of the severity or presence of some disease state. More generally, these are the molecules which like used as an indicator for a particular disease state, or they actually indicate us about a particular physiological state. There are several types of molecules such as low density lipoprotein, LDL, cholesterol, fluctuation in blood pressure can also be a biomarker. P53 genes, MMP, these all molecules, they can act as a biomarker for a different types of diseases. For example, LDL and cholesterol can be a biomarker for obesity, and blood pressure for BP related disorders. P53 and MMPs like matrix metalloproteinases, they can be indicate us about the tumorous growth. Okay, so broadly, biomarker can be of 4 types. They can be of genomic, like gene based. They can be of transcriptomic types, like if we are able to detect the mRNA level, and they can be of proteomics, like like P53 protein, MMPs, enzymes. They are a proteomic biomarkers, and metabolomics. For example, we if we wanted to detect like glucose or certain metabolites, lactate, then they can indicate us about the particular type of metabolic disorders. So all these type of biomarkers, if we can detect their level more precisely, and if their level fluctuates, say for example, normal glucose level is considered as a 80 to 100 milligram per dl in a fasting condition, and if it fluctuates, it goes below it, it indicates the hypoglycemic condition, and if it increase, it generally indicates about the hyperglycemic condition, or which is a major indication of diabetes. So these biomarkers above or below their normal range, they indicates a particular type of disease condition. So, as my ongoing research work is focused on the cancer, and we all know that the cancer is the leading cause of death in the person, and especially breast cancer is the leading cause of death among females worldwide. Conventionally, breast cancer can be detected using mammography, ultrasound, CT scan, MRI, tissue biopsy. But these techniques, as I said, they are costly and costly. So our research is mainly focused on developing a biosensor which can detect biomarkers more precisely in selective and sensitive manner, and which can overcome the disadvantage of the conventional technology. Like they can be handy, and our biosensor can be handy, and they can be used at a point of care mode settings. And since since these biosensors, they are quite handy, they can be miniaturized and they can be used for detection of more than one biomarker simultaneously, uh, like in a multiplexed manner. So nowadays their market actually, owing to their potential benefits over the conventional techniques, their market is going to increase, and it is expected that till 2034 it would be around 127.4 USD billion. So, what are, like I already explained you about the biorecognition elements. If we used antibody as our biorecognition element and we immobilize it over our electrode, then the sensor is known as immunosensor. And if we use aptamers, which is a synthetic nucleotides stretch of a synthetic nucleotide, then the sensor is known as, the fabricated sensor is known as the aptasensor. There is actually a broader view why, like conventional biosensors used antibodies or enzymes as a biorecognition elements, but nowadays people are using alternative of these enzymes and antibodies, such as aptamers or MIPs, molecularly imprinted polymers. These are actually, we can say them synthetic antibodies. They mimic the exact role of antibodies or enzymes, like they are specific for their analytes, and they provide specificity, and simultaneously they overcome the drawbacks of conventional BREs, like in this tabular form I have mentioned few of the advantages or drawbacks of conventional BREs, such as antibodies. They are natural molecules, but they are bulky. They have around 150 to 180 kilodalton molecular weight. Their synthesis takes times. They takes almost several months, and antibodies, they are temperature or pH sensitive. They can lose their activity over once the pH or temperature changed. However, our synthetic BREs like aptamers or MIPs, they are stable over wide range of temperature and pH. They can be designed for wide range of target. However, our antibodies, they are specific, and they are actually immunogenic. They can, once it can insert it inside the body, if simultaneous, if contamination happens, they can causes adverse immune reaction, toxic immune reaction. However, aptamers or MIP, in case of aptamers or MIPs, they are no observed toxicity was seen um, and they can be synthesized in vitro, and their synthesis process is cheap, and they can be stored at ambient temperature. Whereas antibodies, they generally need the in vivo system to synthesize, and they are expensive, and generally their storage needs a low temperature. Aptamers can be synthesized, like I will discuss about aptasensors in today's talk. So aptamers, from where aptamers can be generated. Aptamers can be synthesized using SELEX technology, systemic evaluation of enrichment of ligand-based method. Here, here actually we takes the random amplified sequences of around 5 to 50 kDa, and allow these sequences to bind with the target molecule and elude the bound and unbound, and we use the negative selection mode, where the unbound aptamers were further allowed to bind with the target molecule, and the sequences which showed higher affinity towards their target were selected and allowed to further amplify using PCR or RT-PCR method, and these sequences then now purified using positive selection method, and amplified and stored for further usage. So, what are the aptasensors? Basically, aptasensors are nothing but the biosensor, where aptamers is used as a biorecognition elements. And nowadays, lots of companies are dealing with the synthesis of aptamers. One can purchase them directly rather than optimizing the whole selects technology in his or her lab. So there are key market companies dealing with the aptamer. One is the ICMB Texas. You can search this over the web. So this is the overall schematic how we have fabricated Aptasensor in our lab. We generally first electrodeposited the nanomaterial, reduced graphene oxide gold nanocomposite over glassy carbon electrode, and then after electrodeposition, cyclic voltammetric based electrodeposition method, we immobilize this nanomaterial platform using a specific aptamer. Here in this case, I have used the carcinoembryonic antigen specific aptamer. We used EDC NHS coupling reaction for immobilization of this aptamer, and after immobilization of aptamers, we block the non-specific binding using bovine serum albumin, and now our aptasensor is ready, and we have used this aptasensor for detection of CIA, carcinoembryonic antigen, which is a potent biomarker associated with the breast cancer. And you can see this graphical response, where in presence of CIA, when CIA bounds, when CIA is present in the blood serum sample, it lowers the current. Its binding to the biorecognition elements generally decreases the current output, and without CIA we got the full response. So I have electrodeposited the reduced graphene oxide gold nanocomposite using cyclic voltammetric based electrodeposition method, and this electrodeposited nanomaterial we have characterized using various technique. Like you can see the A picture showing the FSM image of electrodeposited material, rGO-Au material, and B is showing its transmission electron microscopic image, and C is its HR time, and D is SAT pattern, which clearly indicates the presence of graphene and gold incorporated, embedded inside it. And we have also performed some UV visible spectroscopy, XRD and FTIR and Raman, to further characterize the synthesis of rGO gold nano composite. So, and after the synthesis of that nanomaterial or electrodeposition of nanomaterial, we have characterized it, and using FTIR, UV, or Raman, TAM, SAM, and we further validated it using electrochemical characterization method, where we actually measured the response at various steps. Like you can see at GCE, that is glassy carbon electrode, in CV based graph showed 45 microampere current. When it is immobilized with rGO gold, the current response enhanced to 66.43 microampere, and when the rGO gold immobilized with aptamer, the slight reduction in current from 66 to 64 microampere was observed, and after blocking of non-specific binding, the current subsequently reduced to the 43 microamperes. The differential pulse voltammetry, you can see the B graph is representing the differential pulse voltammetric experiment. The results of current is in concordance with the cyclic voltammetric results. And at the lower panel, the graph C is the Nyquist plot, and the graph D is the Bott plot for the electrochemical characterization of the same rGO-Au, aptamer, nanocomposite, and fabricated sensor. And these all the values of current actually validated the fabrication of Aptasensor. After successful fabrication of the Aptasensor, we have used this Aptasensor for the detection of CA antigen in PBS and spiked serum sample. In the PBS, this Aptasensor show a linear range of detection from 10 autogram to 100 nanograms. So you can see its sensitivity is very high. It can be, the fabricated sensor is able to sense the presence of analyte as much as low 10 autogram, that is 10 raised to power minus 18 um, gram. So this much level of analyte we can see, and we can detect the alteration, in any alteration of the normal range of CA in the patient serum sample. And the results in the spiked serum sample was pretty good, and you can see we were able to detect the analyte, and the linear range of detection is from 10 nanogram to 100 nanogram. And we have found that the LOD is, the limit of detection is around 1 femtogram for this fabricated Aptasensor. So this is all about some results of the work, ongoing work, and now what are the fabricated sensor, how one can use. And this is just a futuristic pictorial of Aptasensors. Actually, Aptasensors can be used for diagnostics and more sensitive diagnostics of a particular disease, like detection of a particular disease biomarker, would lead to a timely therapeutics and more strategic or smart. One can design the most strategic therapeutics against a particular disorder if we are able to detect the disease in various phases. Like there are one limitation of the ongoing conventional technique is that they fail to detect the disease in early stages, and the diagnosis at the later stages, especially in case of cancer, would lead to the not only high cost of treatment, but also it costs the life of the patient. So early detection of the cancer is must for its timely management and eradication. So in that case, the biosensor can be able to detect the cancer in very early, or we can say that a slight alteration in the normal range of biomarkers will indicate that it will indicate about the disease, and person get alarmed, and it can change the lifestyle by which it can be like the he or she can be avoid this disastrous disease. So there are some futuristic application of aptasensors. They can be connected with the internet of things, internet of medical things. They can be interfaced with artificial intelligence, and smartphone based operation makes these sensor um, um, make these sensing platform wireless, and 3D printing or additive manufacturing can help in their scalable fabrication, and integration with different software platform provides us easy operation, effective data storage, rapid data sharing, and rapid analytics. So, to conclude or summarize the today's talk, I would like to just emphasize over few points. The advantages of aptamers over natural biorecognition elements include ease of synthesis, cost effectiveness, high selectivity, and specificity. And due to these properties, these aptamers they are they actually offers better properties over natural or conventional BREs, and they enhance the efficacy of electrochemical biosensors. The synthesis of reduced graphene oxide gold nanocomposite. In my case, I have confirmed it using FTIR, XRD, TAM, SAM, Raman spectroscopy, and UV visible spectroscopy. And the aptamer immobilized reduced rGO gold Aptasensor actually showed good stability over a period of month. rGO-Au Aptasensor BSA, this is the name of Aptasensor which I have fabricated, showed excellent carcinoembryonic antigen biosensing ability in PBS and real serum sample, and the developed Aptasensor showed a linear range of detection of C antigen from 10 autogram to 100 nanogram, and the present research work led to the development of this Aptasensor for cost effective and rapid detection of SIA biomarker in which is a breast cancer biomarker in patient serum sample. These are few publication from our group, and these are the facilities which is available at CSIR EMPRI, and I would like to acknowledge director CSIR EMPRI for supporting my research work. And this is my group. I would like to thank my mentor Dr. Raju Khan and my group member who have supported me in my research endeavor. Okay, thank you so much. If you have any question, you can ask.
Biomarkers and how they works, and ma'am had given a very simple example of detection of glucose, what could be the chemical reaction behind that glucose detection itself, and how we can use the nanomaterial for the development of the biosensors, right? So we have some questions, ma'am. So let us take the questions. Okay, the first one is, what are the reference and working electrodes and how do they work?
Working electrode, as I already like showed, working electrode can be of let me okay. Working electrode can be of glassy carbon electrode, gold or silver, and these are the electrode over which we actually fabricate our sensor. Okay, we immobilize our we actually mount our nanomaterial over these electrode. So since this is the electrode where we are going to immobilize our biorecognition element, so it is known as the working electrode. And the reference electrode is AgAgCl. We use this electrode as a reference to monitor the current response. Actually, in case of electrochemical biosensor, we are monitoring the current now. So you can see this here I have shown one picture. Here, this is the three electrode system, where one is the counter electrode, the one is another is the working electrode, and the third one is the reference electrode. Reference electrode, generally we prefer to use the AgAgCl electrode. So the current generated, I think you read it about these electrode system in electrochemistry part, might be in 11th or 12th, where these electrode, when connected with potential state, they generally able to detect the change in current. Okay, so I hope I have cleared your doubt.
Yes, ma'am. The next question is, what is the difference between the aptamers and the biomarkers? Is there any difference in these two?
Yeah, they are the two different things. Actually, aptamers, they are specific to biomarkers, and biomarkers are the one which are associated with the disease. For example, in case of diabetes, glucose is a biomarker. It belongs to the metabolomic class of biomarker. We wanted to detect the glucose. So if we wanted to detect the glucose, we should choose a aptamer which is specific to that glucose. That means that aptamer is able to capture the glucose which is present in the sample, right?
Okay. Okay. The next question is, how to select fabrication for aptamers?
Okay, like if you wanted to develop a diagnostic diagnostic against a particular disease. For example, say for example, I have fabricated the Aptasensor for breast cancer detection. Okay, here I choose a biomarker SIA. One can choose her to human epidermal growth factor receptor EGFR, and PSA, or IL-6, IL-8 inflammatory biomarkers. And you have to just design your aptamer based on your biomarker of interest, and the aptamer should be specific for that biomarker, and you have to immobilize that aptamer over your fabricated sensor. Okay, so if you are asking about the aptamer, it should be specific against the target. And if you are asking about the synthesis process of aptamer, you can synthesize it using the SELEX method.
Okay, I think that was the answer. Next question. We have any other questions? Okay. How can we detect the antibody using aptasensors?
Okay, so this is something interesting. If antibodies, they are, as I already said, that they are bulky molecules around 150 to 190 kDa molecular weight, and one have to design an aptamer. It is a bulky molecule, so you have to have that type of aptamer which can be able to bind a part of this antibody, since it's a big molecule. Okay, so in that case, antibodies comprises generally heavy chains and light chain. You can design an aptamer which can bind with a particular type of light chain associated with the antibody, and then you can be able to detect that antibody in the sample.
Okay, the next question is, can we use the stainless steel conducting plates coated with active electrode material and use it as working electrode?
If these plates are coated, then you can use, because people are using ITO, indium tin oxide coated glass slides, as a active electrode. So coated one can be used. However, the feasibility of this system you have to check first, and then only try it.
Okay. The screen printed electrode system needs less analyte, but while using the classic three electrode system for initial development, we need more volume. Will there be any impact on sensitivity and accuracy?
uh, Yeah, this is another good question. In screen printed electrode, definitely we are using the sample volume in microliters. But first, before moving towards the screen printed electrode system, we have to optimize the process in three electrode system setup. Once our process get optimized in a bigger volume, like 500 microliter, we are using a small cell here, where we can able to detect the analyte in 500 microliter total sample volume. And then we slowly transfer this, like optimize this, and then transfer this platform over the screen printed electrode. And although we have to perform the rigorous experimental part to optimize the whole process, but it is feasible. People are doing this. Hello, Dr. Sakshi, you are not audible.
Thank you so much, ma'am, for answering all the questions. It was very informative and beautifully explained session. And thank you so much once again for giving your time. And thank you, audience, our viewers, and stay tuned with us for next upcoming sessions with our subsequent webinar series. So thank you so much. Thank you, everyone. Thank you.
Thank you so much.